Hey everybody. I hope that you guys are having a good start to your Monday. Today is the second. There's a couple of things that are worth noting. Um, One is that you have a quiz that's available. That quiz went up today at 12 p.m. and there are 10 questions. It just covers the material from last week. Um, this In addition to that, if I remember correctly, today at 11:59 p.m. is when your first problem set is due. If it's turned in tomorrow or any time after 11:59 p.m., there will be a 10% deduction. If it's turned in within 48 hours after, so more than 24 but less than 48 hours after the due date, then there will be a 20% point deduction. But then after that, it will not be available, or you will not be able to turn it in. Complete that problem set, post it up, and submit it through our Canvas site. Um, it has to be a PDF. What I would recommend doing if you are working on paper or something like that, take a picture of what you've done, copy and paste that picture into a Word document or a Google Doc, and then export that as a PDF. If you run into problems with your submission, for instance, if you run into the problem of, oh, I only have I took a picture with my phone. Well, if chances are, if you're using an iPhone, the image file is an HEIC, which Canvas does not like. So you're going to run into a problem. So make sure that you have a PDF. That's my point. Make sure that you are submitting a PDF and you're ready with that PDF. If you have any issues with preparing a PDF, please let me know. I'll hang around for a little bit after class. Whenever I say please let me know, that does not mean at, 10 p.m. tonight, email me and say, hey, I'm having technical issues. That means like, let's get it taken care of now. Not this minute, after class now. Then let's see. A week from today, we're going to have our first exam. On Thursday, you have your second quiz. You will have the answers from problem set number one on Wednesday. You will have the answers from problem set number two, I believe, on Sunday. Depends on, I believe that's, yeah, Sunday, I think. And then I'll have office hours on Monday before the exam. If you are going to be taking your exams online, please contact me. If you're going to be taking them in person, then you do not need to contact me. Just go to, I believe it's Chem 100. Um, Other than that, though, we're going to go ahead and get cracking. Um, I don't say this, I just so you guys know, I always have a tendency to be very like careful with my words. And I kind of like apologize in advance. And sometimes that ends up sounding weird, like what I'm saying right now. Um, but I've got office hours at 12 p.m. Monday, Tuesday, and Thursday. Nobody was in the office hours today. I say that just as like, hey, if you have questions, there's spots. I'm not saying that with a like, no one was in office hours today. I don't want you to take that away or take that as my message, because I want you guys to feel comfortable coming and asking questions. Um, and with that in mind, I'm going to go ahead and switch over to share my screen. And whenever anyone has questions, feel free to add them to the chat. And let's see, is this right? Yeah, that's good. Nope, there we go. Um, so I'm going to switch to my pen, get that going, move my chat window right over here. Then that display. I have two monitors just so I can see everything. And yes, that is a flex. Actually, I think three monitors is a flex. Two is, I think, kind of like you have a job with a computer. Okay, so what we're going to be talking about today is, and if you look at what we've got on the Canvas page, I've actually got two PowerPoint presentations. Um, and then if you look at tomorrow's, there's one PowerPoint presentation. So I've got protein structure today. And the two files are primary and secondary, or it's protein structure one and two. And then the other file is protein structure three and four. So one and two is your primary and secondary structure. Three and four is your tertiary and quaternary structure. Then tomorrow we're going to be getting into actual protein functions. Now, effectively, That's 3 days worth of material. I'm going to try to get as far as we can into protein structure 3 and 4 today. If we don't make it all the way through, then we're just going to pick up with it tomorrow. My aim is that by Wednesday, um, we'll have covered protein structure and protein function of hemoglobin and myoglobin. So it might seem like, oh my gosh, this is a lot. It is, but this is all with an effort to get to enzymes sooner rather than later. Enzymes, we're not tackling those until Thursday. If we get done with some of this stuff, like if we, if today is as efficient as I'm hoping that it is, great. Then we'll tackle some hemoglobin and myoglobin tomorrow. That will get us to enzymes on Wednesday a little bit early. Um, if we don't, that's no big deal whatsoever. Okay, so whenever we talk about proteins, which um, I always kind of think about proteins as your agents of biological function. They're the things that do something. Genetic or your genome stores genetic information, carries genetic information generation to generation, but a protein is going to do something. And I find that to be like it's almost like your destination, your goal is to make a protein. Now that's oversimplifying. And as we know with the central dogma of molecular biology, that's also kind of inaccurate. Um, but I think that it's a good way to, if you're to put proteins into context, they do something. Now, just a second. Nope, didn't happen. Um, so proteins, there's a couple of different levels of organization. And think about these as levels of complexity. So there is your primary structure. And this term, primary structure, is also the amino acid sequence. So this is read in an N to C termini. Just like when we were talking about peptides, we read a peptide sequence where we look at an amino acid in the N to C direction. Your secondary structure, well, it's a little bit more complicated. You have your amino acid structure, but then what does that thing look like in terms of does it move? Does it fold? Does it have any kind of wrinkles to it? It does. And your secondary structure captures some of those. And more appropriately, though, it captures the what are known as local structures. Your tertiary structures is your third level of complexity, and your quaternary structure is your fourth level of complexity. Now, the forces or the types of interactions that stabilize these, your primary structure is stabilized by a peptide bond or peptide bonds, which are covalent bonds. Your secondary structure is stabilized by backbone hydrogen bonding. Your tertiary is stabilized by disulfide bonds and all of your intermolecular forces. And your quaternary structure is the same thing. Now, I always like to use analogies because it helps me understand information a little bit better. The way that I think about this is I always like to think about imagine that you're living in an apartment complex or you live in a neighborhood or something like that. Um, from let's say that you live on a cul-de-sac. And this is your house, this is your neighbors, this is your other neighbors, and here's the other people who live. Now I'm just drawing doodles. Here's all of the other people who are connected within the neighborhood. They live on your street on this cul-de-sac, and here's our cul-de-sac, and there's what UPS turns around and leaves. Your primary structure is basically your way of saying, oh, well, I live next to, my name is X and I live next to Y. And how are we connected? Well, we're on the same street. The connection between us is a covalent bond. I also like to waffle back and forth between an analogy and the actual pertinent information. Okay, so your secondary structure. Well, if you're describing to someone your primary structure, all that you're going to say is, well, I live next to person Y. That's my neighbor. Your secondary structure. would be where you say, okay, well, I live next to Y because that's my primary structure. That's the sequence of my neighborhood. And then, actually, here I'm going to try to draw it a little bit. These lines that I'm drawing are the streets. So here's another street, and here's that street connecting. Okay. You enter the neighborhood here. And when you enter the neighborhood, you can go down this street, and then you can take a right, and that's where you're going to see your house. If you had taken a left immediately, or a right immediately, and gone this way, well, you would have gone down a different street, and then you would have found another cul-de-sac. And then you're going to see there's other houses there. One, two, three, four. A secondary structure is going to say, oh, well, I live next to Y, established by my primary structure, but then house number four is behind me. So we're kind of close to each other, but are we close when it comes to, or what would it be? We're close how the pigeon flies, but are we close based on a walk on the road? Not particularly. Does that make sense? It's kind of a description of, yeah, we're all in the same thing, but here's kind of our organization. Your tertiary structure is going to take an even larger view. It's going to look at where your house is, where your amino acid house is, and this entire subdivision in which you live. Your quaternary structure is going to take an even farther view. And that's going to say, oh, well, you live in, because all subdivisions have the same name. I'll go with Hidden Valley because that's a salad dressing, not a neighborhood, but it's probably also a neighborhood somewhere. Oh, you live in Hidden Valley? That's fantastic. That's a great neighborhood. You know what? Hidden Valley, as our protein subunit, is next to Wuthering Heights. Also, not a neighborhood name, instead a book. So those two neighborhoods, those two subdivisions, Hidden Valley and Wuthering Heights, are right next to each other. They make up a part of that entire town. And that entire town is the quaternary structure of the protein. Now, I'm going to try to revisit this explanation as we get into tertiary and quaternary structure in the event that right now you're like, I don't follow. For the time being, what I want you to know is primary structure is adjacent amino acids, and that is, well, covalent bonds and peptide bonds are going to stabilize that. Secondary structure, hydrogen bonds are going to stabilize that, and those are your local structures. So, where are different amino acids relative to you? Okay, so primary structure, it's read from the N to C termini. Covalent bonds are the only types of bonds that stabilize this. Now, typically this is read using a one-letter and three-letter codes. We looked at this exact same slide, ARK and ALA, or sorry, alanine, arginine, lysine. Excuse me, we looked at this exact same slide on Friday. The primary structure is just your amino acid sequence. A nice little definition here is that when you have 50 amino acids making up a peptide, or it's typically 50 is the break point, where 50 amino acids are larger is going to be a protein. Less than that is typically referred to as a peptide. I mentioned the other day that's not a hard and fast rule where you're going to embarrass yourself if you say, hey, there's a 75 amino acid peptide. But that's typically our separation. Okay, now what I want you to think about with respect to this is we are just using what we've talked about last week, our pKa's, our PIs, in drawing a peptide. Now, what I want you guys to think about here is what we did on Friday, or one of the things that we did on Friday was I showed you this kind of simple shorthand model for drawing a peptide and calculating the charge or calculating the PI. Now, let's say that we have an amino acid that I'm going to go with, let's go with E for glutamic acid. Now, I have to toggle back and forth a little bit because I'm going to be pulling pKa values from our presentation the other day, but that's really no issue. Let's say that we wanted to look at what is the PI of this. Or if we wanted to, first, what we're going to do is draw phase state at pH of 1. Now, glutamic acid is the single letter code, or is the amino acid that's represented by the single letter code E. Glutamic acid has three ionizable groups, N, C, and R. The N termini, the carboxyl end, or the C termini, and the R group. The pKa's for these groups, I do not have these memorized, but instead I'm looking at a different PowerPoint. 9.47, 2.10, and last but not least is 4.07. Now, what I remember about glutamic acid is that glutamic acid's R group has a carboxylic acid. Now, what that means is whenever I'm looking at this molecule at a pH of 1, my N termini is going to have a positive charge because my pH 1 is less than 9.47. Next group is going to be, I'm going to look at my R group. That has a pKa of 4.07. 1 is less than 4.07. It's important to remember that this is a carboxyl group, and a carboxyl group, when it is protonated, is going to have a charge of 0 because a carboxyl group is going to look like this or this. It can be neutral or it can have a negative charge. At a low pH, at any pH below the pKa value, it's going to have a charge of 0. Okay, now next is going to be where I look at the C termini. Well, the C termini is also going to be a carboxyl group, in which case that's going to have no charge whatsoever. So I'm looking at a pH of 1, the amino acid glutamic acid has a charge of +1. Okay, could I get a number in the chat if you say, okay, cool, I follow you? And a letter if you're like, okay. Now what we're going to do is we're going to take this exact same thing, but we're going to level it up. Now, what I'm going to do is I'm going to go ahead, no, actually, what I'm going to go with is one of the fun things that I've enjoyed in my life as a biochemistry instructor and as a biochemist is to write out words or use, I don't know, just use the single letter codes to spell out peptides. I love my wife, and her initials are KRG. So I'm going to use her initials as a peptide that I'm going to draw, and we're going to calculate a couple of different things about this. We're going to look at the charge at a pH of 1, pH of 7, and let's go last but not least, pH of 13. Okay, what I need to do is I need to get K, the lysine, pKa for the N termini is 8.95. C termini, or sorry, the C termini is going to be 10. Nope, that's not. There we go. 2.18. The R group is 10.53. R is 9.04. 2.01. Last but not least is 12.48. And I'm just looking at the exact same numbers that I gave you in Friday's presentation. Um, Honestly, I will, on questions on your exam and your quiz, I'll give you these values or I'll give you the necessary values that you need. Okay, G. What's the single letter code? What does G stand for? Somebody add it to the chat while I'll write this number out. And if you want to add the three-letter code, that would be just as good. G is glycine. Glutamine is Q. That's the curveball. That's one of the ones where it's like, how did you arrive at Q? And to answer that question, um, I remember as an undergrad being taught the mnemonic, and it's not a mnemonic like spelling out a long word, but instead, the single letter code I was told was, well, think about glutamine. And I was like, okay. And then they said, it is the cutest amino acid. And I said, oh, cute. It's adorable. So glutamine, cutamine. You know, I'm giving you guys this free of charge. Well, not actually. I mean, that's a complicated response that I just said. No added charge. There we go. That's what I'll say. Okay. So what I'm going to do is we're going to write this peptide out. And one of the important parts about this is a little bit of a spoiler. We're not going to use all of these numbers. Now, the reason we're not going to use all of these numbers is because whenever I write this peptide, and I'm writing her initials KRG, N termini to C termini. So KRG. Okay. So effectively, what I've got is right here, that's a K. Then my R goes right there. And then, yeah, there would be another one there. Now, looking at this right here, and specifically the green kind of halo that's on that bond, that's my peptide bond. There's an important thing to know about peptide bonds, and that is they do not have an overall charge. So, since those do not have an overall charge, when it comes to all of the pKa values that we just wrote out, what that means is that for, let's go with lysine, we'll start off with lysine. We do not need this pKa value. That pKa value corresponds to the C termini of this amino acid. Well, that C termini looks a little bit like this when it's a free amino acid. But this is not a free amino acid. This C termini is found within a peptide bond. So I don't need that C termini. Now, looking at the other pKa values, add a number if we need both of those other pKa values for lysine, and add a letter if no, we don't. And let's think about this. We've got the N termini. Well, that's there. The R group, yeah, we'll need that one as well. So we do need both of those other pKa values. Now, if we move on and add a number if you think, okay, cool, I followed it. I follow you why we omitted one, but we kept the other two. Great. So now what we're going to do, we're going to use the exact same rationale that we use to dismiss or not include the C-terminal pKa of lysine, but we're going to apply it to the N-termini of our next amino acid or arginine. Because again, arginine is our second amino acid. The N-termini of that amino acid, well, that's wrapped up in a peptide bond. It's not ionizable anymore. So this 9.04 that I just circled, I don't need that. Then, next up, my R group, well, I still need the R group pKa value. The C termini, I do not need that because again, that will be wrapped up in a peptide bond. So that 2.01, I'm going to omit that. Now, can I get a number if yes, you follow? And can I get a letter if wait, rewind? Okay. So what we're looking at is for every amino acid, we're trying to see are those amino acid or are those groups, are they accessible? So let me go and take a here. I'm just going to draw amino acids, backbones, NH3 plus I'm going to draw this at a physiological pH. So at a neutral pH, the C termini of this amino acid is going to be deprotonated. And you know what? That C termini is going to love to react with this right here, an N termini of another amino acid. And when they interact, what's going to happen is we're going to form this bond right here. That's a peptide bond. That's a covalent bond. And what's going to happen is, well, I'm going to switch colors to illustrate this a little bit better, maybe. I'm going to switch to green. What's going to happen is this oxygen and these two hydrogens in one water. Dang it, I ruined my own joke, which wasn't a joke. Two hydrogens and one oxygen, they're going to come together and they're going to make H2O. We've got a condensation reaction that takes place there. Now, the result of that means that this peptide bond right here is resistant to any sort of ionization. So that means whatever the pH is, if the pH goes up, the pH goes down, the peptide bond says, I don't care because I as a peptide bond am not going to gain or lose a proton. So this that I put a circle around here gives me this whole structure right there. So that's the reason that any pKa values corresponding to functional groups that are found within a peptide bond, we are not going to use those. So that's why I omitted the C-terminal group for lysine, my first amino acid, the N-terminal of arginine, my second amino acid, and the C-termini of my second amino acid. And now why I will omit the N-termini of my third amino acid. So effectively, what I've got here, KRG, let me draw this peptide real quick. And I'm not going to draw all the functional groups. Instead, I'm just going to draw the R groups as the single letter codes. So we've got NCC, NCC, NCC. Now, this is the K, this is the R, this is the G. H3 plus, okay, that ionizable group is still, that's protonated. I need this. The R group for lysine, well, that's still accessible and available. The peptide bond, well, that peptide bond blocks those two pKa values. So this atom or this functional group and this functional group are no longer an amino group and no longer a carboxyl group. So they are not ionizable, which is why I got rid of those pKa values. I'm going to get rid of this one right here as well. And then this one right here, which leaves behind, last but not least, my C-terminal pKa for my last amino acid. I'm keeping that one. Can I get a number if you follow that now? And a letter if I'm gonna rewatch this. Okay. So what we've done is we've gone through and looked at all the pKa values for this, and we've got a total of 4 pKa values. So this molecule, how would you describe this as an acid? Okay, well, is it a with 4 pKa values, it has 4 ionizable groups, 4 hydrogens. So effectively, this is a tetraprotic acid. Now it seems a little bit weird, but we've assembled this whole molecule. It's a tetraprotic acid because of the fact that it has 4 pKa values. The titration curve for this is going to look a little bit like this. pH on my y-axis, this is my 2.35, then I've got 8.95, 10.53, 12.48. So I got something that looks a little bit like this. Could that have been drawn better? No. Now, what I want you to do is I want you to look at the possible charges for this molecule. We got to think about the nature of the different R groups. So at a pH of 1, which is what I'm going to draw down here, I'm going to put my little box around KRG. My N termini. That N termini is an amino group. It's going to be positively charged at a pH of 1. Okay. My lysine R group. Well, that is a functional group that has a nitrogen that's positively charged at a pH of 1. Same thing for arginine. Arginine and lysine are both polar positive amino acids. So those will both harbor positive charges at a pH of 1. Glycine, on the other hand, if we look down here at G, glycine is a diprotic amino acid. It does not have an ionizable R group. It will not have an ionizable R group. So I'm going to put an O here, and I'm going to leave that there ultimately. Last but not least is my C termini. So this is a place where glycine actually contributed. We've got a carboxyl group. And what is the charge of that carboxyl group? 0, because we're still at a pH of 1, which 1 is below 2.35. Okay, so all told, at a pH of 1, this molecule will have a charge of +3. Now, what we're going to do right now is a couple of things. We're going to identify the possible charges that it can have. We know that it can be +3. I'm going to draw an equilibrium arrow. And above that equilibrium arrow, I'm going to write the lowest pKa, 2.35. Let's imagine, oh, could this molecule have a charge of -3? We're going to find out. I'm going to put a check mark by the 2.35 because I'm done with that. And whenever I deprotonate that group, it's going to go from a +3 to a +2. Then I'm going to deprotonate my next group, which is going to have a pKa, my next lowest pKa, which is, okay, 12.48, 10.35, 8.95, 8.95 is it. And whenever I deprotonate, that's going to give me a +1. 2 pKa values accounted for. Next is my 10.53. That's deprotonated. I got a molecule with a charge of 0. Check mark. Last but not least is 12.48. And that's going to have a charge of -1 whenever I deprotonate that last group. So to answer the question of could this molecule have a pKa, or sorry, could this molecule have an overall charge of -3? No, it cannot. The next question, the next kind of development from there, does anyone want to guess what it is? What would be the next question? Given the information that we've got with our pKs, maybe at the top of the screen, starts with a P and ends with an I. Calculate the PI. I knew you guys were thinking it, and I saw a couple of people doing this, which is the I'm moving towards my keyboard. So I apologize for interrupting that. But calculate the PI. Now, while I'm talking and just kind of talking, I know that someone's going to calculate the PI. So I'm going to give somebody the space to do that while I'm just talking. But I'm also plugging this into my calculator. I've got my number. Now I'm going to divide by 2. And I'm going to get a PI equal to 11.51. So if I were to put this peptide into a solution with a pH of 11.51, what would the overall charge of my molecule be? 0. Okay. What would the overall charge of the most common form of my molecule be if I put it in a solution with a pH of 11.6? 11.6 is higher than 11.5. The predominant form is going to have a charge of -1 because I went above. Or, oh, crap. No, I'm wrong. I mixed up PI and PKA. I apologize. You guys are right. Don't listen to me on that. I apologize. You're absolutely right. The predominant form is going to be zero. We'd have to get pretty far away from that. Okay. How does everyone feel about calculating the charge or the PI of a molecule? Add a number to the chat if you feel good about it, and add a letter if no, you need to rewatch this. Okay. Okay. Well, I'm going to get this posted to our Canvas page after class. It's 2:35, so we're going to go ahead and proceed. So this is an important part of looking and kind of developing because we looked at individual amino acids. Amino acids assemble or are used to assemble peptides and proteins. Now, what we were just looking at when we did these things like calculating the PI and calculating the charge, that's effectively analyzing the primary structure of a peptide. Now, the secondary structure is commonly referred to as local structures. This is stabilized by hydrogen bonding. Now, where is that hydrogen bonding coming from? Well, it's coming from amide hydrogens and carbonyl oxygens. So if we jump back for a second, I want to point out those to you. An amide hydrogen, well, it'd be this right here. And a carbonyl oxygen, well, this is a carbonyl oxygen. But these local structures are not formed by a hydrogen bond between X and Y, but rather whenever you have something that's considerably longer. So anytime you see a structure like this, what I think is really important to do is look at it and say, okay, what am I looking at? This right here, N, that's an amide nitrogen. C, that's my alpha carbon. C, that is my carbonyl carbon. This that I'm drawing an arrow to is a carbonyl oxygen. And what do you see right below that carbonyl oxygen? I'm going to switch colors so I can highlight it a little bit better. What I see is, go with blue, I see a dashed line. A dashed line that goes down to, what is this atom? It might take, I'm going to see this motion. You're absolutely right. It's a hydrogen. So when we talk about backbone hydrogen bonding, we're referencing, well, this carbonyl oxygen is a part of the backbone of my peptide. Then this hydrogen right here, is it a part of the same backbone? Answer a number if yes, and a letter if no, you don't think that it is. A number if it's the same part or if it's part of the same backbone. Well, let's think about this for a second. NCC. NCC. Well, those are all atoms making up a backbone. And I'm going to switch to a black marker and I'm going to draw effectively my backbone. So this oxygen right here and this hydrogen are connecting different parts of the polypeptide backbone through those different atoms. So an amide hydrogen and a carbonyl oxygen will make that hydrogen bonding. Now, there are patterns that exist. And what I mean by that is, well, just as I showed you the peptide KRG, when you talk about a protein that's, 200 amino acids long, that protein has formed through evolution where it's this specific sequence. Are there patterns in protein sequences? Absolutely. Are those patterns something that people have analyzed and said, oh, you know what? Whenever I see this pattern, I can make a prediction. Yeah, absolutely. So patterns exist and secondary structures can actually be predicted. So it's not every single carbonyl oxygen and every single amide hydrogen that are involved in forming a hydrogen bond that looks like this, but there are some that do. Now, amino acid side chains or R groups. When I talk about R groups, I'm referring to like K, R, G, the individual amino acids. They can influence if a structure is formed, but they are not seen as contributors to secondary structures. Okay, so there's typically a very kind of direct question. Um, Maybe on my exams, maybe on other exams that you take, what type of force stabilizes the primary structure of a protein? Covalent bonds. What type of force stabilizes the secondary structure of proteins? Hydrogen bonding, backbone hydrogen bonding more specifically. Now, moving forward, I'm going to try to just use the term proteins as opposed to saying proteins and peptides, just to, save myself the stumbling. Okay, now secondary structure. What I want you to notice about this is, and I think that it's always important to identify different parts of a peptide bond. So the thing that I always do is I always want to kind of find my equator. I want to find out, okay, here's my nitrogen, alpha carbon, carbonyl. Nitrogen, alpha carbon, carbonyl. Nitrogen, alpha carbon, carbonyl. Whenever I do that, it helps me kind of keep track of ultimately where are my different R groups. So then I can say, oh, I can read this sequence. For instance, when you look at this over here, this is my N termini. And I'm going to look at my first alpha carbon. On my alpha carbon, or sorry, on every single alpha carbon, you always have a hydrogen. The other bond goes to the functional group. Well, in this case, it goes to a hydrogen. Well, that's indicative of one amino acid. That amino acid is characterized by the single letter code G for glycine. If I continue scanning along this peptide, okay, here's my second nitrogen. Here's my first peptide bond, my second nitrogen. Here is my alpha carbon. I've got my hydrogen, check. And then I've got, oh man, a ring. The ring indicates phenylalanine, which is represented by the single letter code F. After that, alpha carbon. So whenever you get more prolific at and you do more practice with this, and this is where I would recommend going to PepDraw, where you can then look at pepdraw.com. I said go to PepDraw as if it's a store or something, but go to pepdraw.com and you can plug something in and read that and say, okay, well, where are my alpha carbons? This is an alpha carbon. So I'm looking for a hydrogen and then functional group. Okay. Then next one, functional group, alpha carbon, functional group. And getting the practice of looking at these peptides and identifying what the different R groups are. Now, in addition to that, one of the things that I want you to notice is and be able to do is to look at a molecule like this and make an approximation of what the pH that this is in. Now, how would you do that? That's a great question. You basically are just looking for functional groups to see if they're protonated or deprotonated. On the C terminal end, my carboxy termini is deprotonated. Now, the carboxy termini of almost every amino acid is roughly 2. I'm going to always give you the numbers, but whenever I see that this is deprotonated, my first thought is, oh, well, since that group is deprotonated and that has a -1 charge, that means that my pH is going to be above the pKa for this group. So that's where I'd be able to say, okay, well, the pH of the solution that this molecule is in is, let's call it 2.1, because that's where I'm guessing my pKa for this is 2. Let's say, hypothetically, if my pKa was 2, then I could say, yeah, my pH is 2.1. Is my pH also possibly 14? Well, based on this alone, yeah, because 14 is larger than 2.1. But do I want to boldly say it's 14 without looking at any other part of the molecule? No. That's whenever I want to look at my N termini. my N termini of my peptide. I want to look at my bookends. My bookend on the C termini said, okay, we'll go with 2.1. The N termini says, well, this pKa is at least about 9. So that tells me since that group is protonated, my pKa being 2 and 9, my pH is at least 2.1, but no more than, let's say, 8.99. Now, as you look through more and more parts of this molecule, you'd say, oh, well, that's an aspartic acid, and that's protonated. An aspartic acid with a pKa, again, you are not going to be asked to memorize this. That's at about 3.4. So my pH is basically above 2.1 and below 3.4. So that would be our approximation based on this. Now, one of the things that I also want you to recognize, and this is a detail provided at the end, R groups alternate from for space. Um, they're just represented as one's going up, one's going down in kind of an alternating pattern. Okay, now the next thing is peptide bonds. We kind of talked about them in terms of they're very rigid. They have about 60% double bond character. So that peptide bond is kind of strong. Now, has anyone ever add a number to the chat if you've ever heard the phrase protein folding? So I know that I mentioned it last week, and you've probably heard it in a couple of different contexts, but protein folding or misfolding is a pretty important area of research. Protein folding, well, what that effectively means is a protein takes on a three-dimensional shape. Now, that gets us into our tertiary and quaternary structure, but a protein takes on this three-dimensional shape, and it kind of looks like, for the most part, a glob. We'll talk about classification of proteins. They're either globular or they're fibrous. Globular proteins are generally kind of spherical, um, but it's almost like I always like to think about a ball of clay. Not one of those ball of clays that you get in elementary school where you're like, ooh, look at that, it's perfectly spherical. Instead, the kind that your art teacher throws to you after they've like ripped a chunk off, where you're like, oh man, there's like finger parts over here. Oh, this is just it takes up a lot of space. Now, how do we get from a linear molecule like what we see here to something that's a big glob? And the answer is we have our peptide bonds that are very rigid and they don't allow for much flexibility. But then we've got our pi or our phi and psi angles that allow for a lot of flexibility. Now, with that in mind, when we look at a peptide bond like this, or when we look at a peptide as a molecule like this, I'm going to highlight that this is our N termini and this is our C termini. So I want to put a little star by each of your alpha carbons. So, or not a star, I'll do a dot. Yeah, there we go. Alpha carbon, alpha carbon, alpha carbon, and last but not least, alpha carbon. So each one of those alpha carbons has a hydrogen coming off of it, as well as the R group. Now, when you think about, and I always like to draw mine NCC, NCC. Here's my alpha carbons. On this plot, you have a peptide bond, which is right here. But then you have two other bonds adjacent to your alpha carbon. Now, on the left-hand side of your alpha carbon, which is this bond right here, that N to C bond, that is known as a phi bond or a phi angle. So this right here is also a phi. This right here is a psi angle or a psi bond. That is also a psi angle or a psi bond. Now I bring these up because your peptide bonds, they're not going to move very much, but your phi and psi angles will move and give you the ability to take this linear molecule and get it into this globular shape. Now, this is known as a Ramachandran plot. A Ramachandran plot shows The complementary phi and psi bond angles in a peptide. The shaded regions here on this Ramachandran plot show the favorable bond angles that a phi angle and a psi angle can take on. Now, one of the ways that I always try to think about this is, and it works decently in my mind, but I always think about my hands. And I'll use my hands as, okay, so let's say that my right hand, which is this one, and my left hand is this one, do they have some limiting factors? Yeah, they do. They're attached to my body. I'm happy about that, but that does limit the rotation that they can have. Does everyone follow me there? So think about my right hand as my phi angle and my left hand as my psi angle. Okay, so these two bonds can rotate in certain directions. And what I'm going to show you is my right hand doing what I'll call a 90-degree rotation. Boom, 90. 180, doable. Okay, now I'm going to reset. And this just shows my lack of flexibility because now I'm trying to rotate my thumb towards you, which would effectively be a negative 90-degree rotation. And as you can see, I cannot really do that. Again, because it's connected to my body. My body being the peptide bond. Does everyone follow that? Okay. Same thing with my left hand, which is going to represent one of the other, or the phi or psi angle. Well, I can rotate this way. I can easily do a negative, or I can easily do a 180, but I can't do a negative 180. Does that make sense? So effectively, these two rotations are limited, or these two bonds, they have some flexibility, but they are limited by the rigidity and the inflexibility of the peptide bond. And a Ramachandran plot shows you the favorable combinations. And whenever you're thinking like, well, favorable to what? Well, the energetically favorable rotation. Okay. Now, this is all the setup for what are the local structures. One local structure, sorry, go ahead.
So sorry, I don't want to interrupt, but I just want to ask a quick question. Can you go back to where you drew out the backbone and like showed where you put the phi and the psi bond?
This right here,
Yes, sorry. And then like it's like that so I'm familiar with the symbols. The one with the circle through the middle, which one is that?
that is phi. And then this one, it kind of looks like a pitchfork of sorts. If I was in a Greek life, I'd probably be able to draw that better. That's a psi angle, or that's psi, PSI.
Okay, thank you so much. I just wanted to make sure I had
Sure,
that
thanks
clear
for
as we move
asking.
forward.
Okay. Okay, so um those phi and psi angles can rotate, and that's what ultimately gives rise or gives way to something like this. This is an alpha helix. An alpha helix is a secondary structure. Now, a alpha helix as a secondary structure, we know it's going to be stabilized by hydrogen bonds between an amide nitrogen or an amide hydrogen and a carbonyl oxygen. Hydrogen, oxygen. This helix is commonly referred to as a right-handed helix. It has a cylindrical shape, and different parts of the helix are on top of one another. Now, when you think right-handed helix, what gives? Well, that's brought up and that's addressed because there are some helices that have a left-handed configuration. And you might think, well, yes, you said, okay, I get there's right and left, but what's the significance of that? The significance of that is basically how that molecule kind of folds together in a particular structure. And a right-handed helix is defined by using your right hand, your thumb is going to be going through the central axis of that helix. And your fingers are effectively the peptide as it winds up. I always kind of think about a right-handed helix as winding up and kind of toward me, as opposed to a left-handed helix, which is going in the opposite direction. Now, I would say that right-handed helix definition a little bit more of a kind of trivial information at this point. Now, They are cylindrically shaped, and just as these things, the way that I always kind of think about it is I like to think about a spiral staircase. If you imagine a spiral staircase that has, 10 rotations, at some point, you could be, let's say you and your friend are walking up a spiral staircase, and your friend goes up basically one flight, and you continue going until you're standing basically right on top of them. In an alpha helix, the distance between you is 5.4 angstroms. So one complete rotation is 5.4 angstroms. Now, a fair question for that is, well, in this alpha helical spiral staircase, we need 3.6 amino acids to make that rotation. So it's about 3.6 amino acids to cover 5.4 angstroms. Angstrom is represented by that symbol right there. Now, to illustrate that point, what I always like to do is I like to point out this position right here is directly below this position right here. So that one and that one. This X that I should probably change to something brighter. That is 5.4 angstroms from that. And there are, let's see, if this is an N termini right here, here's a nitrogen, here's a nitrogen, and then here's a nitrogen. So we have 3.6 amino acids that are required to make that complete rotation. Now, one of the questions that I like to ask relates to that, where I'll give you an example like this, where we say an average alpha helix is about 10 amino acids. Okay. Now, with that information, if one alpha helix, an alpha helix, is 10 AA, how many complete rotations does that have? You got to vote for two. I would agree with that because 3.6 plus 3.6 is 7.2. And there you go. 7.2 amino acids will make up two rotations. This is the one where people, whenever I've asked that question in the past, people will give responses like, 2 point whatever. They'll do the actual math they'll do that finite math and they'll say it's 2.6. And then I just get kind of frustrated with myself for asking that question. But this is kind of the rationale for that question. Now, another sort of question that I like to ask is you have a protein that is 150. Oh, I've got it. Okay. Just a sec. Yeah, we'll just use these. Let's go with this. How tall or long is an alpha helix with 110 amino acids? There's 110 amino acids. Well, how are we going to do this? What we can do is we can say 110 amino acids, 3.6 amino acids per rotation. So 110 divided by 3.6, it's going to be 30.6. What is that? That tells me I have 30.6 rotations. Well, where do we go from here? We know that one rotation is 5.4 angstroms. So if the question is, how tall is this? 30.5 times 5.4. No, 165 angstroms. Can I get a number in the chat if you follow that? Okay. So these are some questions that you should anticipate and be able to answer these sorts of questions. Um, then, just kind of contorting it in a couple of different ways. One other way is something like this. How many amino acids are required for an alpha helix to span a 40 angstrom lipid bilayer? Okay, so this tells you it must be 40 angstroms. So then how many rotations is that going to require? Well, it's not exactly, but roughly 8 rotations. And then for each rotation, you have, I'm going to round up here, roughly 4 amino acids. So we're looking at 4 times 8, 32. Yeah, roughly 32 amino acids would do that. Okay, so I'm going to jump back for a second because one of the things that people have concluded over the years is that the carbonyl oxygen is hydrogen bonded to the amide hydrogen of the fourth amino acid away. So what that means is if we have something like NCC, 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 we're looking at this nitrogen to something like this carbon. So that's the network that effectively we're building up there. Now, that is going to be a repeating thing because this structure right here, I'm going to ask 1, 2, 3, 4. We got 5 hydrogen bonds there. Every successive, basically, part of the backbone is going to bond with another part of the backbone. And this is all going to make this tangled web known as an alpha helix. Okay. Now, this next thing, actually, I'm going to pause and ask, does anyone have any questions about the alpha helix or any of the bonds that we've talked about so far today? All right. So we'll proceed from here. A helical wheel, if you ever hear that term, it's really just a valuable tool for imagining that you're looking down an alpha helix. It's a visual for kind of illustrating where different R groups are. So amino acid number 1 in this peptide, well, that is basically the bottom of the spiral staircase. Right above that, or kind of above it and shifted a little bit, is amino acid number 5, then number 8, number 12. The point that I want you to take away from this is that these groups are going to, or sorry, these atoms, they form this helix, and although like amino acid number 1, if you use my X's as individual amino acids, linearly, amino acid number 1 is pretty far from amino acid number 8. But whenever it comes to this linear molecule here, existing in a solution in a three-dimensional space, well, then 8 is like right on top of it. So that's what a helical wheel is trying to illustrate, is how we translate this molecule from a linear reality to kind of a two-dimensional reality. Okay, so an alpha helix is one of your major and very important secondary structures. Another secondary structure is known as a beta sheet, or sometimes you'll hear them referred to as a beta strand. Beta sheets and beta strands are also stabilized by hydrogen bonds. Secondary structures, what are they all stabilized by? Backbone hydrogen bonds. By the end of this class, whenever you hear secondary structures are stabilized by ... it's going to be just like the muscle memory of what is a mole? 6.02 times 10 to the 23rd. It's just going to be like a zombie type of effect. Secondary structures stabilized by backbone hydrogen bonds. Tertiary structures stabilized by R group interactions. Primary structures stabilized by covalent interactions. So the hydrogen bonds are going to stabilize a secondary structure They are going to stabilize your beta sheets. One of the things that's important that we referenced previously this semester is these bonds right here, these hydrogen bonds here, 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 those are going to be the most strong compared to these right here and here because those atoms are directly across from one another. Those hydrogen bonds are linear, they are straight. These groups are 180 degrees away from one another, whereas these right here, well, there's a little bit of an angle to them. The ones that are directly across from one another are known as anti-parallel, and parallel are your weaker beta sheets. Sorry. So anti-parallel stronger than parallel due to the positioning where their hydrogen bonds are going to be straight or angled. Now, beta sheets, they have different strengths, which we just talked about. But what these things can do is, let's look at this one right here on the left-hand side. This is an N termini going to, well, a C termini. But this is all in one single peptide or one protein. So this could be like, let's imagine that this is amino acid number 35. This might be amino acid number 742. And to illustrate that, imagine that I'm writing these X's out in a straight line. This is 1, 2, 3, 4. And I'd continue on to get to 35. I'm making one complete molecule, and then I go all the way to make 742. So these can be massive molecules and they can be very complicated, which is going to lead to wild and crazy structures like these right here on the right-hand side. The ones that these molecules or these structures that are on the left and right, the difference between them is it's just illustrating them in 3D space versus this is a 2D model of it, 2D, and then these are 3D. Now, with that said, these beta sheet structures are common for, has anyone ever heard of GFP? Go fishing protein. Not really. It's green fluorescent protein. Green fluorescent protein is a fantastically valuable protein for lots of cell biology work. It's enabled researchers to pinpoint where proteins are in a cell. Like, oh, well, here's this protein that has this function. And oh, when it goes to the mitochondria, suddenly the cells begin to die. GFP is the sort of protein that helps people do that. That's what GFP stands for green fluorescent protein. Structurally, green fluorescent protein is a bunch of beta sheets. It's commonly known as a beta barrel protein, and it's just a bunch of beta sheets that have folded up to make what, well, effectively looks like a cylinder or a barrel. Okay. Now, with that, alpha helix, beta sheets, those are the important ones. Those are the biggest rascals when it comes to secondary structures. They're stabilized by backbone hydrogen bonds. There are other secondary structures, but they're a little bit less popular. Think of alpha helices and beta sheets as basically the, I don't know, like the Ernie and Bert of protein structures, whereas your other structures, like your reverse turns, are kind of like Snuffleupagus. Yeah, he was on Sesame Street, just wasn't many people's favorite. He was there. A reverse turn is an example of a secondary structural element. It's just less common. Um, and in terms of the number of amino acids that are involved. A reverse turn it's basically just where a protein is going in one direction or the peptide is going in one direction, and then there's an abrupt turn of it. And so that's where you see different parts of it. Now, secondary structure prediction. As I mentioned earlier, this is something that people are able to predict. Um, if you look on the left-hand side here, I've got your three amino acid or your three-letter amino acid codes. If you look in these two columns, and yes, the third one as well, I'll say that. If you look in these other columns, you'll see a number. The lowest possible number is 0, the highest possible number is 2. And these numbers are what are known as propensity scores. So back in the 70s, there were these two researchers, Chow and Fassman. And these guys were structural biologists. And what they did was they took and wrote a review article. If you're not familiar with a review article, it's kind of like a summary. It's almost like within the field, this is what's known. It's a really valuable update. And what they did was they analyzed all of the different protein structures that had been solved. And they basically went to every single structure and they said, you know what? We're going to find your alpha helices and your beta sheets. And we're going to look at the amino acids that are found in those alpha helices and beta sheets. And then we're going to give them a score. And what they found was that there were some amino acids that had a higher propensity or likelihood to be found in an alpha helix than others. Now, if we look at this chart right here, with that interestingly drawn line there, what you'll see is that there are some, like proline, that has a very low propensity score. That means proline is very unlikely to be found in an alpha helix. If you continue on, proline also has a very low beta sheet or propensity. So proline is unlikely to be found in an alpha helix or a beta sheet compared to something like glutamate or glutamic acid. Glutamic acid just has to do, the names have to do with its ionization state. You'll see that glutamate has a 1.59. By comparison, it's much more likely to be found in an alpha helix. Beta sheet, not very likely. The key of this all is that this is based on research that had already been done. Hey, here are the amino acids that are likely to be found in an alpha helix, and here are the ones that are likely to be found in a beta sheet. Categorization, basically. Now, this might seem trivial or maybe even not interesting, but has anyone ever heard of AlphaFold? Add a number to the chat or a letter if no. A number is yes, and a letter is no. So AlphaFold is a project that I believe Google was the major funder of. And Google's their pursuit, their effort was, hey, let's see if you can predict the 3D structure of a protein. Um, the value of that is if you know what a protein looks like, then you have a sense of, oh, I can design an inhibitor or something that blocks the function of that protein. So whenever you can predict the structure or whenever you know the structure of a protein, that gives you, as a researcher, as a drug designer, something to say, aha, here's what the protein looks like. You know what? I'm going to design a molecule that fits right into the spot. I always like to think of enzymes or proteins as kind of like Pac-Mans or Ms. Pac-Mans. And okay, here's a protein. Oh, wow, there's this cleft right here. There's this opening. You know what? I can design a compound that fits right in here and it locks that protein up in that shape. Proteins have to be able to move in order to carry out functions. And if you can block them, they're stuck. You've developed some sort of therapeutic this way. So I bring that up because that was a project that completed, I believe it was in 2020, that Google and Alphabet, I think is the parent company name, they published their first paper on AlphaFold, where here's what we've come up with, here's all of the proteins that we predicted the shape of, and what that protein looks like. When they published that, that was a pretty big deal because now all of a sudden, people who are, working to design a drug for an anti-cancer therapy, well, they have a protein structure that they can kind of target. And I bring that up because without Chow and Fassman doing this work to predict secondary structure, you wouldn't be able to predict tertiary structure or the 3D shape of a protein. So that's why this is extremely valuable. Now, there are amino acids that are by and large an alpha helix. is most likely to form with an alanine, glutamic acid, methionine, leucine, phenylalanine. Those are more likely to be found in an alpha helix. What's less likely to be found is a proline or a glycine. Histidine and arginine as well are unlikely. Then, for a beta sheet, tryptophan, tyrosine, phenylalanine, methionine, isoleucine, valine, threonine, cysteine. Well, those are likely to be found in a beta sheet. Less likely, proline and glycine. I bring these two up and highlight them because proline and glycine, these are so unlikely to be found in an alpha helix or a beta sheet that these are considered, and I'm going to label this, anytime you see a 2 and then a naught and a UGHT or a 2 degree, that's commonly the notation for secondary. These are secondary structure breakers. If you put a proline in a protein, it's going to disrupt the secondary structure. If you put a glycine, it's also going to more than likely do that. Now, a fair question is why? Because if you think about proline and glycine, they're both nonpolar amino acids, but they're kind of different. A glycine, the R group for a glycine is this right here, a hydrogen. That's definitely different from a proline. A proline is a ring, goes from the alpha carbon back to the N termini. Well, the reason that they kind of do the same thing, but for opposite reasons, proline is considered too rigid. Glycine is considered too flexible. Proline doesn't allow for any sort of rotation, whereas glycine allows for too much. So that's what I want you to take away for those two amino acids. Now, yeah, so tertiary structure of a protein. Okay, before I proceed, does anyone have any questions about secondary structure?
I have a question about the last thing we just went over.
Sure.
So I think like I'm trying to think of like how you would ask things on an exam as we go through. And so would you want us to know like why proline and glycine wouldn't make a good alpha helix or beta sheet? But for the others, would the chart with the propensity be provided if you were asking us about what their affinity would be, I guess?
Yeah, so that's a great perspective to have. And I'm going to try to cut to the chase. When it comes to proline and glycine, think about those simply as secondary structure breakers. So historically, I've asked the question of like these two amino acid or this amino acid is considered a secondary structure breaker, and I'll give you four different options. Alternatively, I might say something like this is considered a secondary structure breaker due to it being considered to be too flexible. So that's why I want these terms too rigid, too flexible to be directly associated with these two amino acids. In terms of propensity scores, I don't want you to commit these to memory. I want you to know the general rule that 0 is the lowest. If it has a 0, if there was some magic new amino acid that had a propensity score of 0, then I'd want you to know, okay, its chances of being found in an alpha helix are 0. A beta sheet, 0. It's not going to be found there. Whereas a 2 is like, oh, well, that's always going to be found there. So this is really more like, do you understand the ranges? And this is an instance where I like to ask about names, actually. So Chow and Fastman, I would be comfortable with those names.
Okay, thank you very much. Because I just wanted to make sure like it was like a conceptual like how to read the chart, not like a memorize the chart.
No, not at all. That's yeah, that would be way too much. But thank you for asking. So, okay. So we've got primary structure, secondary structure. Primary stabilized by covalent bonds, secondary stabilized by backbone hydrogen bonds. And I say that because, or I stress backbone hydrogen bonds because here what we've got is our tertiary structure. Tertiary structure is formed by a single polypeptide or by single polypeptides. This is stabilized by covalent and intermolecular forces. Your intermolecular forces being hydrogen bonding, hydrophobic and electrostatic interactions, as well as ionic interactions. These are found, however, between R groups. So when you think about a when you think about a peptide, at this point, when we look at this is the first time that we've talked about, well, what are the R groups doing? And this right here is really just saying the R groups have an influence on secondary structure. They don't dictate it. They have an influence, or there's correlations. When it comes to tertiary structure, then what we're doing is we're saying, okay, the R groups are doing something. These R groups are interacting with one another. So, for instance, if you said serine, if you remember serine, it has an R group that looks a little bit like this. Or let me redraw that. What type of interaction would you expect serine to participate in? Well, not hydrophobic, unlikely ionic, but it would be a good candidate for hydrogen bonding. It could be a hydrogen bond donor. Likewise, well, this oxygen's got lone pairs of electrons, so it could be a hydrogen bond acceptor. So that R group, that serine R group, is capable of those things. Now, when we look at something like aspartic acid, aspartic acid at a neutral pH, well, is it possible? Is it capable of participating in hydrogen bonding? Yeah, because this oxygen over here has got some lone pairs of electrons. This negative charge, if it sees you know, a sodium ion or something, it's going to be a great candidate for interacting with that. So this would be kind of ionic or hydrogen bonding. Now, what I like to do whenever it comes to this sort of information is I like to show you an amino acid and say, what do you think that this is likely to participate in? Um, I also like you to kind of think generally and say, okay, well, polar positive amino acids, polar negative amino acids. What are they going to participate in? Well, they're polar positives, probably ionic interactions. Polar negatives, again, probably ionic interactions because they have the ability to. Does that mean that that's all that they're going to do? No. It is going to have there is a dependency on, well, what's the pH of that environment? If it's a very acidic environment, maybe it's more likely to do hydrogen bonding than anything else. Um, but this is a place where it's good to go back to and think about, well, what are the different um, groups that I'm talking about? Do I have an ion there? Or do I just have, well, is it just going to be a hydrogen and some lone pairs of electrons? So your tertiary structure is where your R groups interact, but the important part about tertiary structure is this detail right here, a single polypeptide. So if I were to draw this right here, this molecule right here is going to have a primary structure, a secondary structure, and a tertiary structure. And that will be displayed in something like this. Here's yeah, that is my tertiary structure version of this protein right here. Yes, it is a squibbly squibbles there, but that's what it is. Now, I bring that up because I want to contrast single polypeptides with this right here, quaternary structure. Your quaternary structure is formed by two or more polypeptides. Okay. Now, the next statement, stabilized by covalent and intermolecular forces. Hydrogen bonding, hydrophobic interactions, and electrostatic interactions between R groups on different polypeptides. So the same types of interactions that stabilize the tertiary structure will stabilize the quaternary structure. The only difference is, are we starting out with this or this and this? Does that make sense? So one of the things that I want you to understand is that every protein has a tertiary structure, but not every protein has a quaternary structure because some proteins will fold as individuals like this one right here. This one has a random coil on it, but it has only one N termini and one C termini. This is a tertiary structure of a protein. By comparison, this right here, this whole thing right here is a quaternary structure. Multiple polypeptides have folded together to make that whole thing. So this arguably different polypeptides, our group interactions of different polypeptides. Um, that's like that's your important takeaway when it comes to quaternary structure. Tertiary structure is one polypeptide, quaternary structure is two polypeptides. Now, to get into tertiary structure for just a little bit, I think I'm only going to go with like, I don't know, a couple of slides because it's been a long day. We've covered a lot of stuff. Um, Tertiary and quaternary structure, or 3-degree, 3-naught, and 4-naught. R group interactions define tertiary and quaternary structure. Quaternary is R group interactions amongst multiple uh, polypeptides, though. Now, the general organization of proteins. So, in 3 dimensions, proteins are either going to be classified as globular or they are going to be considered filamentous or fibrous. Globular proteins are maybe the proteins that you're a little bit more familiar with without knowing that they are globular proteins. Um, if you're familiar with any of the enzymes in glycolysis, those are globular proteins. They catalyze reactions. They are responsible, hemoglobin, which is responsible for transporting oxygen. That is a globular protein. Filamentous proteins. These are long, extended, and often helical or sheet-like proteins. They have highly repetitive sequences. So what I mean by that is they might have a sequence that would be lysine, glycine, arginine, LGR, LGR, just repeated over and over again for 700 amino acids. So they might have a peptide that, or a protein that might look like that. Now, what they do is they predominantly provide structural support, strength, and flexibility. Some examples: collagen, keratin, actin, and myosin, as well as fibroin, which is the protein that is in silk and spider webs. So, collagen, connective tissue, keratin, your hair, nails, and skin, and myosin, which is what we see in muscle contraction, or actin and myosin. Now, we're going to profile and kind of look at basically 2 different proteins from a functional standpoint. We're going to look at muscle fibers, and then we're going to get into function when it comes to hemoglobin. Um, Muscle fibers, muscles are composed of bundles of single large cells. Muscle fibers are formed by cell fusion and contain multiple nuclei, and each fiber may contain many myofibrils. These are composed of repeating units called sarcomeres, and they're bundles of actin and myosin filaments. And that's where I'm going to stop today. That seems abrupt, but we've been going at it for quite some time now. Now, before I stop my recording, there are some things that are very important to kind of highlight. I would definitely know the peptide, like the alpha helix math. Like how tall is a single rotation? How many amino acids are needed for that single rotation? If you have a polypeptide that's this, or if you have an alpha helix that's this many amino acids long, how many amino acids, or sorry, how long is that alpha helix? Um, Identify the secondary structure breakers. know the different types of interactions that stabilize. Um, and this is a topic that I think is worth like kind of getting your hands dirty, but it's important to keep perspective. Um, if you were to look at, if you were to look at every single university that had a first semester biochemistry class, chances are that the final exams in all those classes, they're going to have the question, which type of interaction stabilizes primary structure? Which type stabilizes secondary or tertiary or quaternary? That's one question out of, let's say their final is 70 questions long. Okay. So that in terms of depth of information, that's not super deep. But is that valuable information? Yeah. That's a good summary. You have an understanding of what stabilizes these things, what stabilizes proteins in general. I just say that because I don't want you to be like, okay, muscles are composed of bundles of single large cells. Okay, he's asking us to know every single sarcoplasm, myofibril, myofilaments. Okay, sarcolemma. Okay, I need to be able to draw this and then describe it, and then I will truly know it. I don't want you to do that sort of thing. And I think that after your quizzes and make sure that you look over your learning objectives as kind of your guide for how do I prepare for this guy's class and this guy's exams. Okay. Does anyone have any questions? If you don't have any questions, I hope you have a wonderful afternoon. I'm going to stop my recording and then I'm going to hang around for a little bit to answer any questions that folks may have. But other than that, I hope you have a great day.